So filtration is completed. Now the filtrate contains some important components. We just go through the components here: glucose, amino acids, sodium, potassium, calcium, magnesium chloride, bicarbonate, urea, creatinine, ketone bodies, low molecular weight proteins, and most importantly, water. All these substances are present. Now it is supposed to move towards proximal convoluted tubule. For our convenience, I have drawn like this. This is the proximal convoluted tubule. These are the linings, the cells which lines the proximal convoluted tubule. And this one, we call it as lumen. We call it as lumen or tube. And these are the cells which are surrounding the, which one? Lumen. These are the cells of the proximal convoluted tubule. Now you can see that one red dots are there between each and every cell. I think you can see this one, red colored one. Are you able to see this red one? They are called as tight junctions. They are called as tight junctions. So they have a specific role. That means between each cell of the proximal convoluted tubule, you can see some tight junctions. And this tight junctions role is different in proximal convoluted tubule and loop of Henle and distal convoluted tubule and as well as collecting duct. So they everywhere you can see this tight junction, but its role is different in every section. That means in every section of the nephron, the role is different. So what is the specialty of this tight junction in proximal convoluted tubule? They are tight junction. The name itself indicates they will not allow any substance, but it is not watertight. But it is not watertight. That means water can move out of this tube into what I can say interstitial fluid or extracellular fluid. So the fluid which is present in this one, this is called as tubular fluid. Listen carefully. This is tubular fluid. That means filtrate or tubular fluid. And the water or fluid which is present inside the cell is called as cellular fluid. And the water or the fluid which is present outside the cell, we are calling it as interstitial fluid or extracellular fluid. Are you able to understand? That is the first point you have to remember. Point number 2. Listen carefully this point. Just now I told that this is the tubular fluid and the fluid or water present inside the cell, and then the water which is present outside the cell, that is called as interstitial fluid or extracellular fluid. Now you have to understand that this Bowman's capsule is present in the cortical region, not in the medullary region. And similarly, this proximal convoluted tubule is present in the cortical region. So that means the interstitial fluid or extracellular fluid which is present in the cortical region is little different, and interstitial fluid or extracellular fluid which is present inside the medulla is different. You have to understand that this tube is entering inside the medulla now. Slowly it is projecting downwards and then it will move towards the medulla. This is right now it is in the cortex and then it moves towards the medulla. That means you heard about a descending loop of Henle and as well as ascending loop of Henle. Actually they are present in the medulla. Then distal convoluted tubule, it is present in the cortex. Then again it will take a U-turn, then collecting duct, it will enter into the medulla. That means part of the nephron is present in the cortex and a part of the nephron which is present in the medulla. So why we are discussing that concept means that we are very much interested about this one, the fluid which is present outside the nephron. The fluid which is present outside the nephron in the cortical region, the composition is different. And the fluid which is present in the medullary region, the composition is different. That you keep it in mind. Later on we will discuss about what difference will come. Now, you just go through another slide here. So again, this is the Bowman's capsule. This is a tube. And you can see that this tubular fluid will be present inside the tube, right? And outside the tube, extracellular fluid or interstitial fluid is present. And these are the linings. That means cells which are lining the tube, right? I selected one cell and have drawn here. You observe here. I have selected one cell here. I have drawn here. And this cell I have drawn here. And this side of the cell, it is in contact with the tubular fluid. And this side of the cell, it is in contact with the interstitial fluid. That means the property of the cell membrane, this side membrane is different, and the property of the cell membrane of this side is little different, not little different, so much difference is there. And the membrane which comes in contact with the tubular fluid is called as apical membrane. And the membrane which comes in contact with the interstitial fluid, we call it as basal membrane or basolateral membrane. I repeat again, I selected one cell, and this cell is in contact with the tubular fluid and as well as interstitial fluid. I have drawn this cell here, and this cell, the cell side, that means the membrane side which is in contact with the tubular fluid, we are calling it as apical membrane, and the membrane side which is in contact with the interstitial fluid is called as basolateral membrane or basal membrane. I think you got the clarity here. Now what is this? This is a blood vessel. That means whatever it is coming out of the tube, it enters into the interstitial fluid and then it moves into the blood circulation. That means it is a blood vessel. We heard about peritubular capillaries. It will enter into the peritubular capillaries. Whether you got the clarity or not, whether you followed the concept up to this extent, everybody,
Yes,
anybody?
sir.
Okay. Now, same diagram, little bit different I have drawn. You just go through this one. Same cell. That means, what is that? Proximal convoluted tubule cell. And this is the apical membrane, and this side is the basolateral membrane, right? And this is a tubular fluid, and as well as this is the interstitial fluid. Don't worry about this one. I will explain you later. So only thing is what you have to understand, this cell separately I have drawn here. And this side of the membrane which comes in contact with the tubular fluid is called as apical membrane. And this side of the membrane which comes in contact with interstitial fluid is basal membrane or basolateral membrane. Of course, some textbooks, they have given a different name for apical membrane. Are you able to understand? You no need to worry about it. This is, you consider that it is apical membrane. It will become easy for you. Okay. Now, the problem starts to you now. Okay. You observe this diagram. In this diagram, actually, you can see that this is the tubular fluid. This side is tubular fluid. This is proximal convoluted tubule, one single cell. And then this one, again and again, I will not repeat. This is the basal membrane. This is the apical membrane, and this is the basal membrane. In the basal membrane, you can see this red colored round structure. Red colored, so many red colored round structures are there. They are called as sodium potassium ATPase. Sodium bar potassium ATPase. What is the role of this sodium potassium ATPase? This is a basic fundamental which you have to understand. So all this, what I can say, nephron, not every part of the nephron. Right now we are talking about proximal convoluted tubule. You just imagine that in the proximal convoluted tubule, total proximal convoluted tubule, each and every cell, it contains this type of ATPases. That means this is a protein. This is a protein which is present on the basal membrane. What is the duty of this protein? What this protein will do, it will transfer sodium from the cell to interstitial fluid, and it will transfer potassium from interstitial fluid to proximal convoluted tubule. I repeat again. So this protein or transporter which transfers 3 sodium from the cell to interstitial fluid, and it transfers 2 potassium from interstitial fluid to which one? Proximal convoluted tubule. This type of protein, don't think that 1 or 2 will be there. N number of proteins will be present on the basal membrane. Continuously what it will do, it will be transferring the sodium from the cell to interstitial fluid, and then potassium from the interstitial fluid to proximal convoluted tubule cell. That means for every 3 sodium, 2 potassium is transferred. But here one problem is there. This protein, that means this ATPase, which is enzyme, actually protein, it requires ATP. And directly it utilizes ATP, it converts the ATP into ADP. I repeat again, whenever it is transferring the sodium and as well as potassium like this particular ATPase, it utilizes, what is that? ATP. Lot of ATP is required for this one. Now, so many doubts should come into your mind. From where this sodium is there? That means this is a cell. You know that cell does not contain lot of amount of sodium. Outside the cell, sodium is okay. Inside the cell, sodium is not okay. Then from where the sodium is coming? That is the first question. Second thing is, like, from where this energy is coming? That is another question. And third, another important thing is, here interstitial fluid, listen carefully, this interstitial fluid is rich in sodium already. It is a lot of amount of sodium is present. That is another point. So you already studied that the movement of the ions are from higher concentration to lower concentration. But it is against the rule. Here already sodium is present. But still this ATPase, what it is trying, it is pushing the sodium from the cell, proximal convoluted tubule cell, into interstitial fluid. That means it is against gradient. That means it is against the rule. Even though sodium is rich here, even though sodium is less here, it is pushing all the sodium from the cell into the interstitial fluid. And then this sodium will get reabsorbed. And this one, this structure is called as sodium potassium ATPase. And actually you should get one more doubt. If you see the diagram, what is this? This green colored one, that I will explain you later. Whether you followed this diagram now, I am telling you, these are the ATPases, energy rich. That means it requires a lot of amount of energy, sodium potassium ATPase. It transfers the sodium from the cell to interstitial fluid. And similarly, for every 3 sodium it is transferring, 2 potassiums will be transferred inside the cell. And it requires energy, and directly, remember, directly it utilizes the ATP. That means directly it will convert the ATP into ADP. So this is called as primary active transport. Primary active transport means where it is against the electric gradient, the sodium is transferred. Second point is it is directly utilizing the ATP. So that's why we call it as primary active transport. Okay, fine. Now we have to give explanation that, like, from where the sodium is coming, where from what you can say, energy is coming. Now you just go through one diagram, then you can understand. I've drawn a diagram here. Just now I told you that sodium is transferred outside and potassium is pumped inside by the sodium potassium ATPases. This is the sodium potassium ATPases. Directly utilizing the ATP. So the answer for your doubt, that means from where the energy is coming, you can see these structures, right? On the lining of the basolateral membrane, not 1 or 2, n number of mitochondria are lying like this. So this mitochondria continuously it is providing ATP to this particular ATPases. So that means if you want to understand the concept of reabsorption, first you have to understand the ATPase. All the problem, that means this initiation starts from ATPases. ATPases require energy, and that's why mitochondria is here. And not 1 or 2. So many mitochondria are lying on the edge of the basolateral membrane. So because this transfer requires ATP, adenosine triphosphate. As usual, this side is called as apical membrane and this side is a basolateral membrane. Remember, only in the basolateral side, this which one ATPase will be there. Don't get confused. So sodium potassium ATPase will not be present in apical. Now you go back to original point. Just now I told you that these are the tight junctions, right? So why tight junctions are required? Tight junctions are very important that by chance, not exactly, by chance, maybe this protein ATPase may shift from this side and may enter this side. To prevent that one, so this tight junctions will be there. So this tight junctions will not allow the movement of this particular ATPase to the another side of the membrane. So that's why this tight junctions are extremely important. Now, I think you got the clarity about this particular concept. Now let us go to another slide. Yeah. Now the question, what is that? You just go through the slide. I told you that glucose, amino acids, sodium, potassium, calcium, magnesium, like that, so many are there. When so many number of substances are there, which are suppose some are going to get reabsorbed 100%, some they are getting absorbed partially, some they never get reabsorbed, everything will get eliminated. For example, you heard about creatinine. Creatinine 100% almost it will get eliminated from the body. That means in the form of urine. But actually you have to understand that glucose and as well as amino acid, they will not get eliminated or excreted. 100% they will get reabsorbed into the body. Understood the point or not? So that means how this glucose and amino acid will get reabsorbed. And apart from that, I have written sodium also. Because sodium is easily permeable to membrane and easily it will get filtered, and lot of amount of sodium will be present in the filtrate. Now the game starts now for this one, absorption. Now first one, reabsorption. Now you observe carefully. This is Bowman's capsule and these are the proximal convoluted tubule. And for our convenience, I have drawn this particular cell little bit larger in size. Don't think that this one is larger, this one is smaller, not like that. For our convenience, I have drawn like that. Now you know that this is the basal membrane and sodium potassium ATPase is there, and continuously it is pushing the sodium out and potassium inside, right? Now how the glucose is reabsorbed? You can see that I selected the first cell. I will observe this one. This is very important. So that means glucose reabsorption is the first priority. It starts immediately after the Bowman's capsule. That means you can see that first 2-3 cells, immediately the glucose will get reabsorbed. Now you observe that sodium from the which one tubular fluid, it is moving inside the cell. And then glucose is moving from the tubular fluid into the cell. Now how it is happening? This is one transporter. Again, the green colored one is transporter. So what type of transporter? Now you just use some common sense here. Here the sodium content in this particular cell is very poor. Why it is poor means sodium is more outside this particular area. Sodium is not, it's not actually much more number. This transporter continuously it is pumping the sodium out of this cell. That means whatever the sodium is coming, immediately it is pumping. It requires this particular pump, sodium potassium ATPase. It requires sodium here. So here sodium is there. Here sodium is not there. So somehow or other this sodium should be pumped inside this cell. So for example, this particular sodium and glucose transporter, this particular transporter, very easily it will push the sodium from tube to cell. But the problem is this sodium cannot travel alone. So while traveling, it also collects the glucose and it pushes into the cell. That means whenever sodium is moving here, and the glucose also will move inside the cell. But here one interesting thing is there. Sodium is moving this side, then some logic is there because the concentration of the sodium is very high here and concentration of the sodium is less here. Obviously, the sodium is moving from higher concentration to lower concentration, nothing special. Definitely it is possible. But it cannot go into the cell very easily. Why? Because it is highly positively charged. Crossing the membrane like that, as such, it is not possible because it will not get dissolved in the lipid membrane. This is a plasma membrane. Even though we call it as apical membrane, this is also cell membrane. So that's why sodium requires one transporter. So this sodium is traveling from here to here. So there is nothing special about it. Just by diffusion, secondary by diffusion, it is moving from the tubular fluid to cell. And how this glucose is moving? That is another question. Because the glucose concentration here initially will be very high. Very rapidly, more number of glucose molecules are pushed from the tubular fluid into the cell. Whenever the sodium is entering, it will collect the glucose and it will push inside the cell. Then slowly and steadily, the glucose concentration in the tubular fluid will become less. Then again, you should get one doubt. When the glucose concentration becomes less in the tube, how come the glucose can be pushed into the cell? Why? Because rule is simple. So any molecule should move from the higher concentration to lower concentration. Rapidly, when glucose is moving from the tubular fluid to cell, then very less number of glucose is present in the tubular fluid. How the glucose can move? That is a speciality. This sodium, even single molecule of glucose also it will not allow. All the glucose molecules will be pushed into the cell. That means 100% glucose will be what you can say reabsorbed here. Everything will be pushed here with the help of this particular which one transporter. But this glucose movement from the tube to cell is not actually what you can say passive. This requires one particular special transporter. This transporter is providing that one. So you should get one doubt. From where this transporter is getting energy? That is another question. The energy required for this particular transporter to push sodium and as well as glucose. Then again you should get one doubt. Why energy is required? Why? Because the glucose is moving against the gradient. Sodium is moving in favorable condition, no doubt about it, from higher concentration to lower concentration. But glucose is not moving from higher to lower. It is moving from lower to higher in the second phase. So that's why it requires energy. Whatever the energy required for this particular transporter, it is provided by ATPase. What about ATPase? Sodium potassium ATPase. Why this sodium potassium ATPase is providing energy to this particular transporter? Because this sodium potassium ATPase, it wants a sodium. It requires sodium. To get the sodium, it provides some energy to this particular transporter. Thereby, it will drag the sodium from the tubular fluid into the cell. But sodium cannot travel alone. It will take the glucose along with it and then it will push to the cell. Then this glucose, what will happen? It will move from the cell it will move out of the cell by diffusion. From what I can say, gaps will be there. From the diffusion, it will move out of the cell, enters into the blood circulation. This is about the reabsorption of glucose 100%. Remember the point, 100%. But when I say 100%, it is not actually truly 100%. Why? Because certain limitations will be there. For example, if you consume 1 kg of glucose or sugar or something, if you are expecting all the glucose will be reabsorbed into the body, it is practically impossible. So one limit will be there. So up to certain limit, it will transfer the 100% of glucose from the tubular fluid to cell. For example, 180 milligrams per deciliter or 180 milligrams per 100 ml. So that much only is the capacity, maximum capacity. Above that, so the sodium cannot pump the glucose from the tubular fluid into cell, and then glucose will start to get excreted out from the body. So this is the problem. So that means within the limit, just because sodium is transporting the glucose from the tubular fluid to cell, it doesn't mean that you can consume lot of amount of sugar. You can't do like that. Why? Because two problems are there. One is the totals. Don't think that sodium job is just to transfer the glucose from this side of the membrane to this side of the membrane. Apart from glucose, sodium will help other molecules also, other, what I can say, transfer of other molecules, transportation of other molecules. Don't think that sodium job is just to push the glucose from the tubular fluid to cell. So many molecules it has to transfer. So when glucose molecules are rich in number or too much in number, then automatically the sodium cannot, what I can say, transfer other molecules, or otherwise it can transfer other molecules, it cannot transfer the glucose molecules. Finally, glucose will start to get excreted from the body, glucosuria. Second problem is this glucose also, excess amount of glucose continuously if you are consuming or if you are a diabetic. I told you that this is a membrane. Do you remember glomerular? You heard about glomerular membrane. So this glomerular membrane will become very thick. So thereby, what I can say, filtration of the glucose also will become problematic condition. So still I have not explained anything about this one. You just remember, keep it, remember this one. So now I think you understood the concept of what is that reabsorption of glucose. It is 100%. Again, I am telling, I am not talking anything about sodium. Still, I have never said that it is 100%. Only glucose reabsorption is 100%. This glucose reabsorption in the earliest stage of proximal convoluted tubule, it will get reabsorbed. It will not go to the end stage of proximal convoluted tubule. Very rapidly, it will get reabsorbed. That's why I have drawn in the beginning. This is about the glucose reabsorption. Next, we are going to discuss about amino acid reabsorption. This reabsorption also 100%. Again, same story. You can see that sodium is moving from the tubular fluid to cell, and it is drawing the amino acid from the tubular fluid to cell. Same principle what we discussed in the glucose. The same principle is applied here, and then very fast the amino acid will be collected, and then it is reabsorbed. Same passive diffusion, sorry, diffusion, simple diffusion, it will move out of the cell. That means it will be reabsorbed 100%, no doubt about it. Again, you should get one doubt. Is it really 100%? No, it is 100%. And again, answer is it is not 100%. If you are in limit, 100% of amino acid is reabsorbed. No, you cross the limit. Consider you consume lot of amount of proteinaceous food, lot of amount of chicken or mutton you consumed. Then obviously you can understand that lot of amount of amino acid will be present in the blood. So if it is meat is very heavy, then you can't expect that all these amino acid can be reabsorbed that much efficiently. So it becomes difficult, quite difficult to reabsorb each and every amino acid. Then some amount of the amino acid may get excreted out from the body. That is also possibility will be there. So same mechanism what we discussed in the glucose, the same mechanism is applied to the amino acid also, because it is moving against the gradient, because it is moving from the lower concentration to higher concentration. That means it requires energy, and that energy is provided by sodium potassium ATPases. So now amino acid is over. So this also I have drawn in the first cell. That means first few cells, 1, 2, 3 cells, a few cells. That means in the beginning of the proximal convoluted tubule, this glucose and amino acid will get reabsorbed. Now we are going for a next complicated one. So we discussed like which one? Glucose and amino acid. The most problematic, interesting one is which one? Chloride. Now we discuss about the chloride here. You can see that I have not selected the first few cells. I have selected the last cell. Are you able to understand? Can you see the difference? For example, here I have drawn that sodium and glucose are transferred here, sodium and amino acid are reabsorbed here, but here I have not discussed in the beginning, I am discussing at the end. That means what is the meaning is, the first priority of the kidney is reabsorption of glucose and as well as amino acid. But whenever sodium is moving, it is understood that chloride will accompany it, no doubt about it, sodium chloride. So how this chloride is accompanying it? Now you can see that, so different mechanisms are there by which the chloride will get reabsorbed into the body. You just observe very carefully. First one. Here you can see one transporter. Now instead of showing, you just go through this particular transporter. Sodium is moving out. You can see that the sodium is moving out. That means from the tube it is going into the cell. Along with what you can see along with sodium, chloride also is moving. This is something different here. It is not like what we discussed in glucose and as well as amino acid. This is a co-transporter. Sodium also is moving, chloride also moving. Both are moving, what you can say, like in favorable condition. So it is a co-transporter. This transporter name is called as sodium chloride co-transporter. Sodium chloride co-transporter. That means chloride can be reabsorbed. This is the first method. Sodium along with the chloride, it will move from the tubular fluid to cell. This is method number one. One more method is there. Chloride can also move from the tube into the interstitial fluid by this tight junction. Can you see this tight junction? Through the tight junction, the chloride can move from the tubular fluid into the interstitial fluid. That is the second method. One more method is there by which the chloride can move. You see the water here. Observe the water. What actually water is doing? When a lot of amount of sodium, chloride, everything is moving from the tubular fluid into the cell or interstitial fluid, and automatically what will happen? Water always follows sodium. Then when sodium is moving out along with the glucose, chloride, amino acid, then water will start to rush from the tubular fluid into the interstitial fluid. How? One is, one is the water, it will move through the cell. That means it enters inside the cell and it moves out of the cell. Second method is water can move between the cell. So for example, you can see that between the two cells, through the tight junction, it is moving. So that's why I told you that tight junctions are different in every part of the nephron. Different parts of the nephron, tight junction meaning is different. So proximal, listen carefully, proximal convoluted tubule tight junction is not watertight, is not watertight. It is permeable to water. Water can freely move from the tube to interstitial fluid through tight junction. I am telling you that only in proximal convoluted tubule. Similarly, water can also move from the tubular fluid into the cell, and from the cell it can move into the interstitial fluid. But here one point you have to understand here. When water is moving, it will rush actually. It will rush from the tube to interstitial fluid. While moving, what it will do? It will collect some sodium, it will collect some chloride, it will collect some potassium, etc., etc. It will drag the sodium, potassium, chloride, etc., etc. from tubular region to interstitial region. This is called as reabsorption by drag. By the water literally it will drag the sodium, potassium, chloride. That means especially chloride can be dragged from the tubular area to interstitial area. So this is about the which one chloride explanation. And last but not least in this particular slide, I told you that I have not given explanation to this one. Now you listen carefully this one. I just now explained that like sodium potassium ATPase is there, and this sodium potassium ATPase continuously it is pumping out the sodium outside and bringing the potassium inside. But this potassium continuously, if it is entering inside the cell, what will happen? The positive charge inside the cell will become too much. Potassium, potassium, potassium. Lot of amount of potassium will be loaded inside the cell. So potassium cannot manage that. What you can say, the cell may burst because of excess amount of potassium. So what one facility is there? You can see that one leakage channel is there. This is called leakage channel. The potassium which enters inside, again it will go out of the cell through leakage channel. You can see that. Potassium will enter inside with the sodium potassium ATPase, no doubt about it. But later what happens immediately, The potassium again it will escape from the cell and it will move out of the cell through leakage channel. Listen carefully, this is a leakage channel through which potassium is leaking out of the cell. That means sodium also is pumped out of the cell, potassium also is leaked out of the cell. Then what the cell contains? Rich amount of chloride, very rich amount of chloride. So the charge of this particular cell is minus 70 millivolts because negative ion concentration is very high. So that's why the charge inside the cell is more negative when compared to positive, because sodium also is pushed outside, potassium also is leaked outside, only chloride will be present, lot of amount of chloride is present. So that's why the cell is charges negative, minus 70 millivolts. So up to this extent, it is sufficient today. I think you have to understand that first priority is glucose, amino acid, then next priority is chloride. But definitely chloride also will be absorbed, reabsorbed along with the sodium, because the charge of this particular cell will become negative. Apart from that, water reabsorption also we discussed. Water can be reabsorbed by the transcellular. Transcellular means water moving inside the cell and then moving out of the cell into the interstitial fluid, or paracellular, water moving between the cell in through the tight junctions, that is called as paracellular reabsorption. So this one you have to understand. I hope you understood the concept in a better way. Any questions means you can ask. And one more thing, I know that yesterday class, I think you are not actually convinced by the explanation of which one we discussed about the, what is that? What is the topic? Glomerular filtration rate. So slowly and steadily you can understand glomerular filtration rate. I will repeat that topic again after completion of reabsorption. You no need to worry about it. Whether you followed today's concept or not, you can up to this extent. Whether you followed everything. Any doubts? Any questions, please? At least are you able to understand the concept today? Oh my God.
Yes, sir.
So any
Yes,
questions
sir.
are there, you can ask. Otherwise, after you can go through again, and then if you have any doubts, tomorrow you can ask. Till now, we discussed the normal mechanism by like how it happens in the normal condition. We have not discussed about the diseased condition. First, we will discuss only normal status only. We are not going to discuss anything about the diseased status. Okay. So this point you have to understand. So two things you should not forget. One thing is everything starts because of this one. Which one? Sodium potassium ATPases. This one. This is the fundamental. And you have to understand that if there is a damage to this sodium potassium ATPases, then sodium cannot go out, and sodium cannot go out and potassium cannot come inside. When sodium, if sodium should be available first, if this is damaged, then the sodium cannot enter inside the cell, and sodium cannot enter inside the cell, glucose also cannot enter inside the cell, amino acids also cannot enter inside the cell. Now first what you have to understand, first you have to think about the normal, then you should think about next level is, that means in the pharmacology, pathophysiology, you have to think that what are the factors which will damage this particular sodium potassium ATPases, like drugs like tetracycline. You heard about mercury? Heard about mercury or not? Are you alive, you people? Heard
No,
about
sir.
Yes, sir.
mercury? Heard about mercury?
Yes,
Lead.
Yes, sir.
Yes.
So water contains, like you heard about water, groundwater, you heard about bore water or something, it contains mercury and lead, everything. And apart from that, we consume so many antibiotics like tetracycline, etc. So all these things, they have the tendency to damage this sodium potassium ATPase. That is one issue. And I told you that leak channels will be there, like potassium. What are the factors which will damage? And you also should think that what are the factors which will, and this proteins, what I told you, sodium, potassium, ATPase, ready-made, it will not be available. They should be synthesized. Then you have to think in the next level that what are the factors which promotes the synthesis of sodium, potassium, ATPases, because you require. If it is not available, then automatically there is no use of mitochondria, because mitochondria is meant only to provide energy to this one. Because mitochondria is providing energy to this one, then you require oxygen to produce the energy. You require water, you require food in the form of glucose. So in that way you have to think. That means you have to go back. Not like, you should not think that I will consume diet, I consume water, I consume oxygen, not like that. Back you go. So this is a funda, first starting. You go back. If you want this one, you require mitochondria. If you want mitochondria, that means mitochondria, if it wants to be functional, then you require water, oxygen, and food. That means, so when this is working, automatically you can utilize the glucose, you can utilize water, you can utilize the oxygen. Then it is understood that this is working more. Then lot of amount of carbon dioxide will be produced. Understood the concept or not? So you have to study from the beginning to end. Then again you have to go back from the end to beginning. Then you will get a clear clarity. So we are at the end now. Are you able to understand? This is the excretory system. We are at the end. Then slowly you should go back. So the problem is you require a very strong basics about the biochemistry. I think you have little idea. Electrons transport chain. I think you remember. Do you remember or not? Yes or no? Heard about electron transport chain?
Yes, sir.
Yes, sir.
You have to link it. That means you have to link that electron transport chain and you have to understand the concept. Then it will be a very interesting topic. This topic is interesting, little complicated, I agree, but it is very interesting. It's very important also. So many chapters you are going to study under pharmacology based on this funda. That means sodium, potassium, ATPase, pump. Still not over, still we are in basics. Next class we will discuss in detail. Okay, shall I wind up?